So, but what is systems biology? How can we um, define systems biology? So, Leroy Hood is a very good scientist, one of the main scientists of systems biology. Today, he is really mainly working with medicine to try to find drugs that can treat disease. So he said that a good definition is given from here is that science to the systems biology is a science to discover, model, understand, and project the dynamic relationships between the biological compounds or molecules that make up living organisms. So there are some words very important here. So to discover new things, to model. So it's part of systems biology to have models. Okay, that's why we have this mathematical part of systems biology. So we have models to predict the response. This is the main aim of systems biology. And then understand and project the dynamic. Dynamic is a very key word. It's a key word for system biology. Okay, so understand the dynamic is even more important than understand um, the steady state accumulation of some metabolite or uh, this paper also from Rodrigo Gutierrez, it's also nice to see. In the systems biology, you have the experimental work, which is the lab work. You have this kind of data integration that we can integrate into models or integrate different levels, physiological with biochemical, with molecular, and so on. And then you have modeling, systems modeling. So here you create hypotheses. So you have data that can feed the models. The models create a hypothesis, and the hypotheses are confirmed or not by the experimental work. So if the model is creating nonsense hypothesis, so it means that the model is not right. So you have to fix the model, and this is for the modeling guys is something not really difficult. So uh, the most difficult part here keeps being the experimental work, which demands a lot of time, right? For example, in this situation that we are living now, the systems biology have already created many hypotheses of which compounds can be used to beat coronavirus and which drugs we can use or what kind of vaccines we can have to beat coronavirus. But the difficult thing is the experimental work, especially when you have humans involved. So, because you have a lot of ethics things to follow, and you cannot take a population and test something that you are not really sure that it's gonna work. Okay, because the models can be wrong, right? So if you have a good model, the model you perfectly fit the experimental data. But if you don't have, you're going to have some troubles. So in the systems biology, you keep having the experimental work because we the model is not, we cannot blindly believe in the model. We need some confirmation from the laboratory work. And you can use systems biology from different levels, from molecular, cellular, physiological, organism, and ecological. Actually, systems biology, people sometimes misunderstand that systems biology has to involve these platforms, these omics platforms. No, it is not necessary. So the people from ecology are using these systems philosophy, let's say, since uh, 20, 30 years ago, even more, 40 years ago, they are using this systems way of thinking about science. So you don't need to have large-scale data production to have a systems biology work. Systems biology is more a way how you think and how you interpret the things than just the interpretation of the data, of large-scale data. No, you don't need to have a lot of data to do systems biology. But taking the courses and the research lines that are the basis for systems biology, so you can have, for example, complex systems, the science of networks, the biochemistry, the physiology, computational biology, and so on, molecular biology. You combine all of this and then you have systems biology. Okay, systems biology is huge, it's very complex, so that's why needs a lot of information from many fields. Another definition about complex systems, so biological systems are complex. Okay, plants especially are extremely complex. Okay, so but what is a complex system? We have another good name here. Perhaps no, for sure it's the main name about complex systems and network biology. Albert Laszlo Barabasi. Barabachi, I guess, is the correct way of pronouncing his name. So he said, complex systems are composed by many elements connected to each other by different interactions. So the definition is very simple, right? For a complex system. So you have many elements, for example, proteins, metabolites, genes, that interact with each other in different ways. So this is complex systems. Complex systems are not only biological systems. You have non-biological systems that are also complex. For example, a network of computers in the biochemistry department, it's a kind of complex system. Okay, but in this case, it's not biological. So this is the guy, Albert Laszlo Barabachi. I was preparing this slide and I got curious about how these papers how his papers have been cited. And surprisingly, he has more than 200,000 citations in the total. So this is a huge number of citations. He got, for example, around 16,000 citations per year in the last 7 years, which gives us, for example, 44 citations per day or 1.8 citations per hour. So when he finished this class, he probably had 2 citations. So it's completely crazy to think on this. And he has a H index of 146. It means that he has at least 146 papers that has been cited 146 papers at least. Some of his papers has been cited more than 20,000 times. It's crazy. 20,000 times, it means that 20,000 papers has cited one of his papers. It's insane. He's really good, but to be honest, he's my inspiration. Not because of the productivity, but I didn't know even these numbers so far. I'm reading this guy since I started my biological course. So he's kind of my inspiration. Why he is important for science? Because he was one of the first to take the theory of complex systems that exist since 1946, for example, because in physics they used this theory for a long time, but he transformed these complex systems and network science into biological systems. And how he did this? So complex systems, you can visualize and interpret them by networks. For example, you have here the nodes, which corresponds to the metabolites, and then you have the links, which corresponds to the enzymes, and you can also have, for example, this kind of feedback regulation based on allosteric regulation. For example, one metabolite of the end of the pathway can regulate one step before of the pathway by binding the enzyme and inhibiting or activating them. We have seen this already in the last class about ATP, when ATP is accumulated a lot, so the respiration is blocked. The citrate is also one metabolite that have this feedback regulation. So it means that we can have metabolism. We can visualize and interpret metabolism by using this network organization.